this lesson, we'll look at the fundamental units of life, cells. The specification has the following to say about this: eukaryotes and The specification has a short section at the end listing the key ideas underpinning everything that we are studying. One of these key ideas is that all life is made up of cells. Chemical reactions are what make living things tick. The things which make life distinctive, movement, respiration, sensitivity to their environment, growth, reproduction, excretion and nutrition, they all rely on special molecules and very particular reactions. For life to occur, these special molecules, represented here by moving shapes on the screen, need to be kept together, in association with each other. If they drift apart, then they can't react with each other. So there needs to be a membrane to keep these molecules together. This is the cell membrane. Without a cell membrane, the special chemicals would just drift off. They wouldn't meet and interact to produce the chemistry of life. To interact with the outside world and gain the energy-rich molecules that are needed to sustain life, a cell needs to exchange materials across the cell membrane. So the larger this membrane is compared to the interior of the cell, the quicker this exchange can take place. When we compare the outside surface of a cell to the interior space, we find that smaller cells have more surface for each unit of interior space. They have a larger surface area to volume ratio. So they have more absorbing surface to provide the materials for the interior to make use of. Try working out the ratio of the overall surface area to volume for the two cubes shown here. One made from just one cell, the other, with the same overall volume, made from lots of cells. This increased surface area to volume ratio of smaller cells is why cells are as small as they can be while still containing the chemicals needed for life processes. The first cells to evolve were very small, with all the chemicals of life floating freely within them. These are the prokaryotes. That's the archaea that live in the type of extreme conditions found when the Earth first formed, and the bacteria. Prokaryotic cells have a cell membrane and another supporting layer surrounding this called the cell wall. They also often have a whip-like flagellum to aid movement, but they have very little internal structure and no other membranes. The essential difference between the primitive prokaryotes, that's the archaea and the bacteria, and the more highly evolved eukaryotes, that's everything else, is the presence of a membrane around the genetic material. Prokaryotes lack this membrane. They have a single circular chromosome floating freely within the cytoplasm. The nuclear membrane possessed by eukaryotes fulfills a similar role for the genetic material that the cell membrane does for the rest of the cell. It controls what can enter and what can leave the nucleus. This enhances the ability of the cell to regulate which genes, that's the unit of instructions in the DNA, which genes are being used by a particular cell and which are not. This increased ability to control what we call gene expression allows cells with a nucleus to specialize, and then because of that to work as part of a team, a multicellular organism. Using membranes in this way to provide special conditions in particular regions of the cytoplasm allows eukaryotic cells to create organelles with the ability to perform specialist functions. The presence of these specialized membrane-bound organelles within eukaryotic cells gives the cells an increased range of abilities. But the organelles take up more room, so eukaryotic cells tend to be around 100 times larger than prokaryotic cells. To understand how different cells function, we need to understand what each of the components within the cells do. The cell membrane. Now, as we've already said, all types of living things are made up of cells, and all cells have a cell membrane. The cell membrane forms a barrier to the outside world. It controls what can enter and what can leave the cell. Very small, uncharged molecules such as oxygen and water can move through this barrier relatively freely. But charged ions and larger organic molecules can't squeeze through. Embedded within the cell membrane are some special proteins which span the membrane and provide a controllable pathway for other molecules. By changing the amount of these proteins embedded in the cell membrane, or by controlling their behavior, the cell membrane can regulate what is able to pass into the cell and what cannot. Another key idea mentioned in the specification is that the activity and function of molecules is related to their structure. Many of the proteins embedded within the cell membrane are shaped so that only particular molecules can use them to pass through the membrane. So changing the amount of these proteins embedded within the cell membrane can change how permeable each individual cell is to particular chemicals. The nucleus. Again, as we've already mentioned, the nucleus is only found in eukaryotic cells. The genetic material contained in the nucleus is made of a chemical called DNA, deoxyribonucleic acid, and it's organized into a series of strands called chromosomes. The structure of DNA allows it to store encoded instructions. These instructions, which we call genes, are used to create proteins. By changing which proteins are made and when, the nucleus is able to control the activities of the cell. Prokaryotic cells don't have a nucleus, but they do have DNA in the form of a single circular chromosome floating freely within the cytoplasm. Without a nucleus, prokaryotes have less control over which of the instructions on the DNA are used. But they do have some control, and they can use the instructions on their single chromosome to produce the particular proteins needed as conditions change for the cell. Plasmids. In addition to their single chromosome, prokaryotes have or they often have, some extra DNA in the form of plasmids. Now these are very small circular strands of DNA that replicate independently of the rest of the cell, and they're not always present. When they are present, the very small number of additional genes held on the plasmids can be useful to the main cell under certain particular conditions, such as when antibiotics are present. A rather bizarre feature of plasmids is that bacteria can often swap them with each other, and so transfer the abilities afforded them by the plasmids. Ribosomes. This is where the instructions from the DNA are used to make the proteins used by the cell. Ribosomes are one of the very few structures that are found in all types of cell, both prokaryotes and eukaryotes. Flagellum. These are whip-like structures found on some prokaryotic and some eukaryotic cells. They are used a bit like a propeller to move the cell from place to place. Cell wall. An additional supportive layer of polysaccharide is found outside the cell membrane of many cell types, both prokaryotic and eukaryotic. In plants, the polysaccharide is called cellulose. In yeast, it's called chitin, and in bacteria, it's called peptidoglycan. Animal cells are among the very few cell types that lack a cell wall. A mistake often made by students is to assume cell walls are selectively permeable. They're not. In plants, the cell wall is made of the same sort of material as found in cotton wool. And cotton wool doesn't prevent water flowing through it, but it can be wound into strong rope-like strands. The cell wall of plants is similar to this. It gives strength to the cell, and that allows pressure to build up inside the cell without bursting the very delicate but selectively permeable cell membrane which surrounds the cell. Cytoplasm. This is the gel-like substance found in the interior of the cell. It's not an inert jelly. It's where the chemical reactions of life take place, and in the more highly evolved eukaryotes, it's crisscrossed by a network of membrane pipes and sheets used in the manufacture and transport of the chemicals used by the cell. Mitochondria. This organelle is where aerobic respiration takes place, where the eukaryotic cells release the energy from food molecules. The more energy a cell type uses, the more mitochondria it's likely to have. An interesting feature to note is that mitochondria in our cells are thought to have evolved from separate, free-living organisms that inhabited our cells. The ancestral mitochondria presumably benefited from the food and oxygen molecules found in the cell, and the cell presumably benefited from the energy released by the mitochondria. The arrangement was so successful that mitochondria and the cells they lived within evolved to become so dependent on one another that neither could live without the other. Support for this theory comes from the fact that mitochondria still to this day contain their own DNA, and they replicate independently from the nucleus of the cell. Chloroplasts. This organelle is only found in the photosynthetic cells of plants, the cells above the ground and in the light, never the roots. Chloroplasts contain the green pigment chlorophyll, which has the enzymes needed to absorb light energy and use it to create glucose from carbon dioxide and water. Now, like mitochondria, chloroplasts also contain their own DNA, and they are thought to have evolved from a successful association with another primordial invader. Large permanent vacuole. Vacuoles are just membrane sacs, and they're used for a lot of different purposes in eukaryotes. The large permanent vacuole it's a particularly important vacuole only present in plant cells. Its purpose is to absorb water and increase in size. With just the delicate cell membrane, the cell would burst, but the cell wall prevents this and so causes the cell to form a rigid shape. A good analogy for this is the flexible inner tube of a bicycle tire inside the outer tire. When the inner tube is pumped full of air, the tire prevents it from bursting and holds the wheel in shape. In this analogy, the inner tube is the large permanent vacuole and the tire is the cell wall. Take a moment to look through the summary, then press continue to work through the questions that follow. Don't forget to download the resources for this video from the link in the description, especially the notes template. LearnFix has a lot more to offer than just the videos and the exam questions. The most popular aspect is the set of 12 online learning games we have for each topic. These help you practice your recall of the details. Now when it comes to revision, LearnFix is there to help. After each section of the syllabus, there's a diagnostic quiz to help focus your revision efforts. You can then use a progress report to look back at the details for that particular topic, what went well and what needs more work. We're adding new features every month, so visit us at learnfix.com to find out more.